Okay, so let's get started. So welcome back, everyone. A reminder to start with is that the tutorial classes will begin next week. The first tutorial is a kind of group activity, hands-on activity that's aimed at sort of reinforcing some of the concepts that we learn about in today's lecture. And there'll be a student handout that guides you through the activity in class. And so I'll put that on Canvas next week. And if you can either print it before you come to the tutorial or bring your own device to the tutorial so that you can look at the handout. Okay, so last week we started looking at what the individual cells of our brain are up to. And we saw that the main function or activity that they're engaged in that seems to be important for understanding cognition and behavior is communicating with each other, so sending signals to each other. And so to expand on that, our ability to see relies on signals being sent from our eyes to our brain. Our ability to move depends on signals being sent by neurons in our brain to our muscles. And our ability to think and remember and make decisions and so on all rely on neurons in our brain sending signals to each other. And these signals have two main parts that are worth learning about. They first begin with an electrical signal, which is generated at one end of the axon near the cell body and then travels down the length of the neuron's axon to the synapse. And this electrical signal we're going to look at in detail in today's lecture. And then this electrical signal triggers the release of a chemical signal at the synapse that can then be detected by other neurons. And so that chemical signal is going to be the focus of next week's lecture. These electrical signals are happening all the time. So right now there are billions of neurons in your brain and throughout the nerves in your body that are generating constantly these tiny electrical signals. And the sum effect of all that electrical activity happening inside your body is actually strong enough that it can be detected from the surface of your head, for example. Shown on the slide here is a more direct way of measuring these electrical signals. It's where a device called a microelectrode, which you could think of as being like a little pin or a very thin wire, is inserted into the brain and into a particular neuron. And then it's hooked up to a device that is able to measure changes in voltage that are occurring in that specific neuron over time. To help give you a bit more of an idea of what these electrical signals are and why they're worth learning about, I'm going to briefly outline some famous experiments that were performed in the history of neuroscience. These were done by two researchers at Harvard University, David Hubel and Torsten Wiesel, in the 1960s and 70s, and they won a Nobel Prize for this work. And what they did is they were using a microelectrode, like what we saw on the previous slide. They were inserting this into the brain of cats in order to measure the activity of individual cells in the cortex of the cat. And in particular, they were recording from cells in an area at the back of the brain called the visual cortex. So it's at the back of our brain as well. We have this same kind of area. And this is an area of our cortex that receives signals from our eyes. And so as they were recording the activity from neurons in this area, they would show different images to the cat on a screen in order to figure out what kind of images might produce a response in the neurons that they were recording from. So that's illustrated a bit further here.
The researchers actually listened in to individual nerve cells firing in the anesthetized cat as they presented it with different visual images.
When we started working, Torsten and I, in the late 50s, we set up our first experiments, and they didn't go well because at the beginning we couldn't make the cells fire at all. We'd shine lights all over the screen. and nothing seemed to work. And rather by accident, one day, we were shining small spots, either white spots or black spots, onto the screen, and we found that the black dot seemed to be working in a way that at first we couldn't understand until we found that it was the process of slipping the piece of glass into the projector, which swept a line, a very faint, precise, narrow line across the retina, and every time we did that, we'd get a response.
Okay, so their microelectrode is placed to come in contact with a single cell at a time in the cat's cortex. They're showing the cat these different simple images on screen, these dots and lines and whatnot. And you would have heard this kind of crackling or buzzing sound in the background. And this is actually a representation of when the cell that they're recording from is becoming active. Okay, so they've hooked up their microelectrode to the speakers in the room. And whenever they record a change in voltage of the cell, it produces this crackling sound. So in this way, they can monitor in real time when the cell is becoming active by listening to it as they show the cat different types of images. And that's further illustrated in this video. So what you're seeing in that video is effectively what the cat is seeing. And again, that buzzing sound in the background is when the neuron that they're recording from in the cat's visual cortex is becoming active. And you can see here that this neuron that they're recording from is responding to a particular feature of what the cat is seeing. So it becomes active whenever this spot of light appears in a particular part of the cat's field of view. And there's another similar illustration here. Okay, so here the researcher is waving this object in front of the cat's line of sight, and when the object is in a particular location at a particular distance from the cat, this neuron that they're recording from is becoming active and producing these electrical signals. And so what these experiments illustrated is that there are these cells in the cortex of the cat's brain that are responding to particular things that the cat is seeing. Like spots of light in a particular location or objects in a particular position. And the same is true in our brain as well. So as you look around the room right now, there are many neurons at the back of your brain in the cortex that are becoming active and producing these bursts of electrical activity, like what you hear in the video, in response to the position and the movement of objects around you. And so in a later lecture, we'll talk a bit more about what the significance of this is for understanding the brain's role in our vision. But here I'm just using it as an example, or to give you a bit more of a feel of what these kind of electrical signals are. So this is some of the data from their experiments. What you're looking at here is a recording of changes in voltage of the neuron that they're recording from. And this series of vertical lines or spikes represents a burst of electrical activity in the cell that they're recording from when the cat is viewing a particular kind of stimulus, like a vertical line. If we zoom in on one of those spikes, it would look like this. And so in this plot, we have the voltage of the cell that they're recording from in the y-axis. We have time in the x-axis, and so this is occurring just over a few milliseconds. And this electrical activity of the cell involves these really characteristic fluctuations up and down in the cell's voltage. And this is referred to as an action potential. And this is a key part of the electrical signal that the neuron is generating and what we're going to focus on in today's lecture. So a point that I'm trying to make here is that when we talk about the activity of our brain, most of the time what that refers to is these electrical signals that neurons are producing. So sometimes it's referred to as neurons firing when they produce these electrical signals. And what they are, or the way that we measure them, is as a change in the voltage of the cell that we refer to as the action potential. So you may have come across the amygdala before, this area of the brain that's traditionally been linked to emotion. You may have heard the idea that the amygdala becomes active when we're stressed or afraid, when we feel fear. Later in the course, we'll talk a bit about sleep, and we'll encounter the idea that our cortex becomes more active when we're dreaming compared to when we're in a deeper sleep. And in both of these examples, what we're saying by these areas of the brain becoming more active is that neurons in those areas are producing these bursts of electrical activity, like what you heard in the video, at a greater rate, to a greater extent. Okay, so today we're going to focus on what these electrical signals are, how they're generated in the cell. By the end of the lecture, you should be able to describe the cellular processes that underlie the action potential, as well as something called the resting potential, which is a closely related concept that I'll introduce in a moment. And you should be able to explain how these electrical signals travel down the length of a neuron. So what are these electrical signals? I've described how we can measure them as changes in the voltage of the cell, and that these changes in voltage initially begin at one end of the axon where it meets the cell body, and then they travel down the length of the axon to the synapse. And what these changes in voltage really reflect is the movement of ions between the inside and the outside of the cell. So to unpack what that means, we can first look at the membrane of the cell. So last week we talked about how the cell has the neuron has these three main components. It has a cell body. It has these dendrites, these branch-like protrusions from the cell body, and then it has an axon that it uses to send signals to other neurons or to other organs in our body. And all of these structures have a kind of outer skin or an outer coating, which is the cell membrane. And so we're looking at a kind of schematic of a cross-section of that here. And so the cell membrane keeps the inside of the cell separate from the outside of the cell. It's made up of lipid molecules. So lipids are a category of chemicals that includes fats and oils. And these lipid molecules have a phosphate group attached to them, a particular chemical structure attached to them, and so they're referred to as phospholipid molecules. The cell membrane contains two layers of these molecules, and so it's sometimes also referred to as a phospholipid bilayer. And the important part here for our purposes is that it's this cell membrane that is regulating what's able to enter the cell and exit the cell. In this schematic, again, we're looking at a cross-section of the cell membrane, this lipid bilayer. On the inside of the cell, we have fluid that we refer to as intracellular fluid, and outside the cell, we have extracellular fluid. This fluid is primarily made up of water with various other things that are floating around in it. And so you can think of a cell as being like a little sack of water that's immersed in a larger bath of water. One of the important things that is floating around in the fluid, both inside the cell and outside the cell, are ions. And so ions are atoms that have, or other molecules that have an electric charge. This comes about when the atom loses an electron or gains an electron, and ions can be either positively charged or negatively charged. Some of the ions that are important to understanding the function of neurons are illustrated here. We have sodium ions, Na+, so positively charged ions, chloride ions, Cl-, negatively charged ions. And so you may be familiar with these as NaCl, so table salt that we eat is made up of these ions, and they're also present in our brain. And then potassium ions are represented as K+, and so these are also positively charged ions. If you feel like you could use a primer on what ions and atoms are and electric charge, then the textbook has an appendix that has a little bit of a kind of revision of this. But if you're happy with the idea that atoms can have a positive or negative charge, then that's really most of what you need to know for this course. Okay. An important feature of neurons is that at rest, the inside of the cell is more negatively charged compared to the outside of the cell. And so there are both positive and negatively charged ions outside the cell and inside the cell. But if we added up all of the positive charges inside the cell and all the negative charges, and we compared that to the positive and negative charges that are outside the cell, we would find that overall there's a net negative charge inside the cell and a net positive charge outside the cell. So that electrical charge, that difference in electrical charge between the inside and the outside of the cell, is due to the presence of these charged ions. It can also be described as an electrical gradient, so a difference in electrical charge between the inside and the outside of the cell. And the electrical gradient of the neuron, when it's at rest, when it's not producing these electrical signals, we refer to it as the resting potential. We can measure this in neurons by inserting a microelectrode into the neuron, and what we find is often that the resting potential of neurons is around negative 70 millivolts, as shown on the plot here. And so the negative there signifies that the inside of the cell is more negatively charged than the outside of the cell. The action potential, in contrast, is a change in the voltage of the cell away from the resting potential. So in this figure that we've seen already, we're starting with the resting potential at negative 70 millivolts, and then this fluctuation up and down in the voltage of the cell is what we call the action potential. And this is related to the movement of those ions that we were just talking about across the cell membrane, which changes the electrical gradient. It changes the difference in charge between the inside and the outside of the cell. So first of all, how are ions able to cross the cell membrane? So the cell membrane is represented here again, that lipid bilayer, with the inside of the cell at the bottom, the outside of the cell at the top. And in green and yellow here we have sodium and potassium ions. The lipid bilayer itself is impermeable to these ions. So the sodium-potassium ions can't cross through the layer of lipid molecules directly. But the cell membrane also contains these specialized proteins that are embedded in the lipid bilayer, and these are illustrated here, that are called ion channels, and which can allow ions to move from one side of the membrane to the other. And so these ion channels are like little doorways in the cell membrane. They can be closed at certain times and they don't allow ions to move, or they can be open and then that allows ions to move from one side of the membrane to the other. And then a further detail is that these ion channels tend to be specific to particular ions. So we have sodium ion channels in our brain that allow sodium to move through, but don't allow potassium to move through, for example. And then we have different proteins that are potassium ion channels that allow potassium to move through, but not other ions. And then a further question is, why would these ions move? So why don't they just stay still? Why don't they just float around randomly? And there are two main factors that are explaining why when and in what direction these ions move across the cell membrane. The first is the electrical gradient, so the difference in electrical charge between the inside and outside of the cell. We've just been talking about the resting potential, how at rest the inside of the cell is more negatively charged compared to the outside of the cell. Sodium and potassium ions are both positively charged ions, and opposite electrical charges attract each other. And so this electrical gradient that occurs in the neuron at rest is attracting these ions to move into the cell. So here we have a depiction of that. So both inside the cell and outside the cell, we have these positively charged sodium and potassium ions floating around in the fluid. Overall, the net charge of the cell inside is more negative than the outside, and that negative charge inside will attract the positively charged ions that are outside of the cell and to compel them to move into the cell. The second factor that explains why ions move is called a concentration gradient or a chemical gradient. And this refers to the difference in concentration of these ions inside the cell compared to outside the cell. So at rest, sodium ions are more highly concentrated on the outside of the cell, while potassium ions are more highly concentrated on the inside of the cell. And ions will tend to diffuse from an area where they're more highly concentrated to an area where they're less highly concentrated. So that's illustrated here. For sodium, when the cell is at rest, we have a higher concentration of sodium outside the cell compared to the inside. So both the concentration gradient and the electrical gradient are both encouraging sodium to move into the cell. For potassium, on the other hand, we have a higher concentration of potassium ions inside the cell compared to the outside. So the concentration gradient for potassium is actually working in the opposite direction to the electrical gradient. The concentration gradient is predicting that potassium would diffuse out of the cell, while the electrical gradient is predicting that potassium will be attracted into the cell. So then we can ask, why don't the ions just move? Like if there's more sodium outside the cell and it wants to move into the cell due to the electrical and concentration gradients, why doesn't it just move into the cell and kind of equalize those electrical and concentration gradients? And the first explanation for that is that the sodium ion channels in the cell membrane are closed when the cell is at rest. So sodium wants to move into the cell, but it can't because the cell membrane is impermeable to sodium ions and the ion channels are closed. Another factor is something called the sodium-potassium pump, which is illustrated on the right here. This is a specialized type of ion channel in the cell membrane of the neurons that is working all the time to pump sodium ions out of the cell and pump potassium ions into the cell. And it does this by using energy. So our cells have different sources of energy, and this pump is using that energy in order to pump sodium out of the cell against its concentration gradient. So we can make a distinction between two different types of ion channels, so active and passive ion channels. The passive ion channels are analogous to a gate in a canal, where if the gate is closed, it will block the flow of water. If the gate is open, water can flow, but the direction that it flows will just depend on what other kind of natural forces are there. So water often flows in, or generally flows in rivers because of gravity, and so if the gate's open, then gravity will determine what direction the water flows. And similarly, these passive sodium ion channels, for instance, can be open or closed. If they're open, then what will determine which direction the ions flow through the channel will just depend on these natural forces that are present, the electrical and the concentration gradients that are present. This contrasts with the active ion channels, like the sodium-potassium pump, which are expending energy in order to move the ions against the kind of natural forces that are present, against their concentration and electrical gradients. And so this is analogous to a motorized pump, where it's using energy in the form of electricity or fuel in order to move water against the force of gravity, to pump water up to the top of your apartment building or something like that. So the full picture is illustrated here. So again, we have the cell membrane, the outside of the cell at the top, the inside of the cell at the bottom. At rest, there is a high concentration of sodium outside of the cell, these green balls, and a high concentration of potassium inside of the cell in yellow. The sodium ions are attracted into the cell by the electrical gradient and the concentration gradient, but the sodium ion channels are closed, so the sodium ions are stuck outside of the cell. The sodium-potassium pump is constantly pumping more sodium out of the cell and more potassium into the cell. And at rest, there are some potassium ion channels open, and that allows some of the potassium into the cell that's inside the cell to diffuse out of the cell due to the concentration gradient. But at the same time, potassium is a positively charged ion, and the inside of the cell is negatively charged compared to the outside. And so some potassium that's diffusing out is just sort of coming back in again, attracted by the electrical gradient. So the key learning points here are that at rest, the inside of the neuron is negatively charged compared to the outside. There is a difference in the concentration of sodium and potassium ions on either side of the cell membrane, more sodium outside and more potassium inside. The electrical and chemical gradients suggest that sodium ions should be entering the cell, but they can't because the sodium ion channels are closed. And this state of affairs in the neuron at rest sets the stage for the action potential, for the ability of the neuron to generate these electrical signals. Okay, so are there any questions about this at this stage? Okay, so we can have a break in a moment, but let's do a couple of poll everywhere questions just to check how you're following along. So again, you can scan the QR code on your phone or follow the link in order to put in your response and see what other people are choosing. So the first question is to consider a neuron that's at rest with a resting potential of negative 70 millivolts, signifying that the inside of the cell is more negatively charged than the outside. What do you think will happen if sodium ion channels open in the cell membrane? Do you think the voltage across the cell membrane will become more positive? The voltage will become more negative? Or will the voltage of the cell stay the same? So taking into account what we've learnt about the distribution of ions and the forces acting on ions. Okay, great. So pretty much everyone chose A, that the voltage will become more positive, and that's a correct answer. So we've just been looking at how sodium is this positively charged ion. At rest, there are these forces acting on the ion that are compelling it to move into the cell, and so if the sodium ion channels open, it will move into the cell due to the negative charge inside the cell as well as the concentration gradient. And because it's a positively charged ion, as it moves into the cell, it will make the cell more positively charged inside, which in this case will result in a more positive, or at least less negative, voltage that we're recording from the cell. Okay, the second question is that when a neuron is at rest, there's no net flow of potassium across the cell membrane. Is this because potassium ion channels are shut? Because the sodium-potassium pump is transporting potassium out of the cell to balance the potassium that's diffusing into the cell? Is it because potassium ions leaving the cell due to the chemical gradient are balanced by ions that are entering the cell due to the electrical gradient? Or is it because potassium is unable to leave the cell because it is positively charged and the inside of the cell is negatively charged? Thanks. Okay, so let's get started again. So we've been talking about the resting potential, so this electrical state of the cell when it's not producing electrical signals. And this contrasts with the action potential, which refers to these changes in voltage that occur in the cell when it is that it's able to use as a kind of signal to help communicate with other cells. And this action potential involves a series of different steps, different underlying chemical events that are producing this fluctuation in voltage that you see on the slide here. And I'm going to go through these steps kind of briefly just to give you a general picture of what's happening, and then I'll come back and go through each step a bit more slowly. So we start with the resting potential, the voltage of around negative 70 millivolts, shown on the plot here. To trigger an action potential, there has to be some external stimulus that the neuron receives that will push the voltage of the cell across a certain threshold that will trigger the action potential. And so, in particular, we need what's called a a depolarization of the cell, which in this context is a change in its voltage towards a more positive voltage. And so the action potential is triggered in a neuron when it receives signals across the synapse that it shares with other neurons that cause an initial weak depolarization of the neuron that crosses a certain threshold shown here. And so I won't talk more about that input that the neuron receives to trigger the action potential in today's lecture, but that will be more of a focus of next week's lecture when we talk about the synapse. So if the neuron is depolarized and its voltage crosses the threshold, that will trigger a rapid and large depolarization, which is the start of the action potential, which is this shooting upwards of the voltage to a positive voltage. And that's caused by the opening of something called voltage-gated sodium ion channels that allow sodium ions that are outside of the cell to rush into the cell. And because sodium ions are positively charged, that pushes the voltage up to a positive voltage. And so I'll talk more about what a voltage-gated ion channel is in a moment. The voltage of the cell quickly reaches a positive peak, and then it again rapidly returns back down to a negative voltage. And we call that process repolarization as opposed to depolarization. And that repolarization, that return to a negative voltage, is caused by the sodium ion channels closing, which means that the sodium ions are no longer able to continue entering the cell, and the opening of additional potassium ion channels that allow potassium ions to exit the cell in greater quantities. And as those positive potassium ions exit the cell, it brings the voltage of the cell back down to a negative voltage. What happens is that the voltage actually goes even more negative than the baseline state, the resting potential, and we call that hyperpolarization. And so finally, the final stage of the action potential is this gradual return over a couple of milliseconds from that hyperpolarized state back to the resting potential of around negative 70 millivolts. And that return to the baseline state is caused by these voltage-gated potassium ion channels closing, which limits the flow of potassium out of the cell. Okay, so the key points here are that the action potential is this rapid change in the voltage across the cell membrane, and it reflects the opening and closing of ion channels in the membrane that allow these charged ions to move into the cell and then move out of the cell. So to go back to the start again, when the action potential is triggered, we get this rapid depolarization of the cell membrane. I noted that this is caused by the opening of voltage-gated sodium ion channels. And these are the same sodium ion channels that we were discussing in the context of the resting potential, which are closed at rest. A feature of these channels is that they are voltage-gated, which just means that whether they're open or closed depends on the voltage of the cell. Voltage-gated channels have a threshold, and if the cell's voltage is below that threshold, they will be closed. If the voltage is above that threshold, they will open. And so for the sodium voltage-gated ion channels, the threshold is around negative 55 millivolts here. So when the cell is at rest and it has a resting potential of negative 70 millivolts, that's below the threshold, and the sodium ion channels remain closed. When an external stimulus, signals that the neuron gets from other neurons across the synapse, causes an initial weak depolarization that pushes the voltage of the cell across that threshold, it triggers those sodium ion channels to open and a large amount of sodium to move into the cell, causing this much larger depolarization of the cell. Okay, so that's illustrated here, that at rest we have more sodium outside of the cell compared to inside. We have the electrical and chemical gradients that are compelling sodium to move into the cell. And so when those voltage-gated sodium ion channels open, these positive ions on the outside move into the cell and make the voltage more positive. The next stage is the repolarization, where the cell returns to a negative voltage. And this is caused by those sodium ion channels becoming closing and becoming inactivated, which means that sodium is no longer able to enter the cell and affect its voltage, while at the same time voltage-gated potassium ion channels open, which allows potassium ions to leave the cell. So when we're at the peak of the action potential up here, the state of the cell can be represented something like this, where the positive sodium ions many of them have moved into the cell, making the inside of the cell more positively charged overall compared to the outside of the cell. For potassium ions, we saw that at rest, they're more highly concentrated inside of the cell compared to the outside. And now that the voltage of the cell has flipped to a positive voltage, it means that the electrical gradient is now also attracting potassium ions to move out of the cell, because they're positively charged ions attracted to negative charge, and now the outside of the cell is more negatively charged than the inside. And so when those voltage-gated potassium ion channels open, it allows lots of potassium ions that are inside of the cell to leave the cell, and because they're positively charged, as they leave the cell, it makes it more negatively charged inside, which we're representing as the voltage here going more negative again. Okay, and that whole process only takes a millisecond or so. It happens very, very rapidly. And then for another couple of milliseconds, there's this relatively gradual return from a hyperpolarized state where the cell is more negative inside than it was at rest, back to the resting potential of around negative 70 millivolts. And so that hyperpolarized state is caused by the larger number of potassium ion channels that are open, allowing potassium ions to move out of the cell, making it more negative inside. And those voltage-gated potassium ion channels close, which limits the flow of potassium out of the cell, and that allows the cell to return itself back to its baseline resting potential. Okay, so to sum up, at rest, the sodium ions are mostly outside of the cell and potassium more so inside the cell. During the action potential, we first get this movement of sodium into the cell, illustrated here, followed by this movement of some potassium out of the cell. And then in the meantime, in the background, the sodium-potassium pump is continuing to work to maintain the concentration gradients that occur when the cell is at rest by pumping sodium ions back out of the cell and potassium ions into the cell. And so what that means is that if a neuron undergoes several action potentials, where in each one some potassium enters the cell, then the next one more potassium enters the cell, over time, without the sodium-potassium pump, more and more sodium would build up inside the cell. But because the pump is there, always pumping some sodium out and some potassium in, it helps the cell maintain these concentration gradients that occur at rest. Okay, so the key points here are that the action potential includes a positive spike in the voltage of the neuron that we call depolarization, followed by a rapid return to a negative voltage that we call repolarization. The depolarization is driven by the opening of voltage-gated sodium ion channels and the movement of these positively charged sodium ions into the cell. And the repolarization is driven by the closure of those sodium ion channels together with the opening of voltage-gated potassium ion channels and the movement of potassium ions out of the cell. Okay, so it's that movement of ions that's producing this electrical signal, this change in voltage of the cell. And now there's a question of how is that useful? Like, how does it allow the cell to generate a signal, or how does it use that as a signal? And what happens is that this action potential initially occurs at one end of the axon where it meets the cell body, and we can refer to this as the axon hillock. And then it propagates from that position down the length of the axon to the synapse. Okay, and as it propagates, what we mean by that is that adjacent parts of the cell membrane will undergo that action potential in sequence from one end of the neuron to the other. To illustrate why that occurs, here we're taking a neuron and we're zooming in to a part of its axon near to the cell body. So this is a representation of the cell membrane with the outside of the cell at the top and the inside at the bottom. The action potential will first occur at this part of the membrane in this example. So we have sodium ion, voltage-gated sodium ion channels opening, allowing sodium ions to enter the cell. So the first stage of the action potential. When those sodium ions enter the cell, it means that the concentration of sodium ions at that part of the axon will be higher than it is at adjacent parts of the axon. And these sodium ions are able to diffuse within the cell, within the axon. And that carries some positive charge within the axon to the next segment of the axon, the next segment of the cell membrane. And that positive charge will then cause that initial weak depolarization of the axon that's required to trigger the opening of voltage-gated sodium ion channels, and therefore triggering sort of a new action potential, if you like, to occur at that next part of the axon. And then the sodium ions that enter due to the opening of sodium ion channels at that second part of the axon will also diffuse within the axon and trigger the opening of sodium ion channels in the next part of the axon, and so on. So it's this chain reaction where the influx of sodium at one part of the axon leads to sodium ion channels opening at the next part of the axon, and so on. And that works as a chain reaction from one end of the axon down to the other end. So the key point here is that the action potential propagates along the length of the axon of the neuron due to this chain reaction, where ions that enter one part of the axon during the depolarization phase will trigger the opening of voltage-gated sodium ion channels in adjacent parts of the axon. Another concept that's relevant to understanding how the action potential travels along the axon is something called the refractory period. And this refers to a period after which a neuron has produced an action potential where it's limited in producing another action potential. And this puts a limit on how quickly a neuron can produce these electrical signals. There are two components to this refractory period. So one is called the absolute refractory period, which is a period of 1 millisecond or so following an action potential in which the neuron is unable to produce another action potential. And then there's a slightly longer relative refractory period where following an action potential, it's possible for the neuron to fire another action potential, but just less likely, more difficult. These two different components of the refractory period have different underlying mechanisms. And so for the absolute refractory period, it relates to the inactivation of the voltage-gated sodium ion channels. And so we talked about how when the action potential is triggered, these voltage-gated sodium ion channels open and the voltage of the cell becomes more positive. At that point, the voltage-gated sodium ion channels close and become inactivated for about 1 millisecond or so. And during that period of inactivation, they won't open again regardless of the voltage of the cell. And so that means that it's not possible for the neuron, or at least that part of the axon, to generate another action potential because these voltage-gated sodium ion channels won't open. So with these voltage-gated ion channels, there are 3 different states that they can be in effectively. So here we're looking at the channel at rest when it's closed. We have sodium ions outside the neuron at the top here, and but because at rest the voltage of the cell is below the threshold to open these channels, the sodium ions can't enter the cell. When the cell is depolarized, it triggers these voltage-gated sodium ion channels to open, allowing the sodium ions to enter the cell. And then the sodium ion channels become inactivated, this third state, which means that they're closed. They won't let the sodium ion channels pass, but also it will just stay closed regardless of the voltage of the cell. It stops caring about the voltage of the cell and it just stays closed for a period of 1 millisecond or so. And that's what underlies the absolute refractory period of the neuron. The relative refractory period relates instead to the period of hyperpolarization that the neuron undergoes towards the end of the action potential. So we saw that as the neuron repolarizes with the potassium ions exiting the cell, making it more negative inside, it actually moves down to a more negative voltage than it is at rest. And that more negative voltage means that to produce another action potential, there's a greater distance that the voltage needs to move in order to reach the threshold that will open the voltage-gated sodium ion channels. So the action potential is triggered when those voltage-gated sodium ion channels open. That requires the voltage of the cell to reach a certain threshold, which here is negative 55 millivolts. And so the voltage of the cell is hyperpolarized, even more negative than it is at rest. It means that a stronger stimulus is required in order to trigger the action potential, to reach the threshold that's required to open the voltage-gated sodium ion channels. And next week we'll talk about what that stimulus actually is in more detail when we talk about the synapse, and so this concept will make more sense there then as well. Okay, and so this refractory period, it puts a limit on how quickly neurons can produce action potentials, but it also has a role in preventing the action potential from moving backwards along the axon. It enforces the action potential to move in one particular direction effectively. And so to illustrate what I mean by that, we talked about how the action potential moving along the axon is caused by this diffusion of or the entry of sodium ions into the axon, and how the positive charge that those sodium ions carry can trigger the opening of voltage-gated sodium ion channels in adjacent parts of the axon. And so if you imagine that what we're looking at here is a central part of the axon, in principle, the entry of the sodium ions could trigger the opening of sodium ion channels on either side of that point, such that the action potential would just continuously sort of bounce back and forth, or would sort of go in two directions along the axon. But because of this refractory period, it means that if the action potential has initially been generated at one end of the axon, then the sodium ion channels in that part of the axon, after they've undergone the action potential, will be in their refractory period, so that when the next part of the axon undergoes the action potential, the sodium ions that enter and diffuse to the left in this example will meet sodium ion channels that are in their refractory period, and so will have no effect. It won't trigger the action potential to happen again, whereas when they diffuse in the other direction, along where the action potential hasn't already been, they'll meet ion channels that are not in their refractory period and trigger the action potential to occur in that part of the axon. So this is why the action potential will kind of move consistently from one end of the axon to the other. Okay, so the key point here is that after the action potential, there is a refractory period during which it's either impossible initially, or at least less likely for a bit of a longer period, for the neuron to produce another action potential. Okay, so one final structure we're going to talk about that's relevant to how neurons send these electrical signals is a structure called myelin, which is illustrated in purple here along the left. So it's this insulating sheath that forms around the axon of many neurons in our brain and in our body. And it has the function of effectively insulating the axon and allowing it to send these electrical signals more quickly. So it increases the speed at which these action potentials propagate down from one end of the axon to the other. On the right here, we're looking at a cross-section of an axon. So the axon is this section here, and you can see around it is this myelin sheath. It's made up of these different layers of membrane that have wrapped around the axon. Last week, we talked about glia cells, which are this other category of cells that are found in our brain, apart from neurons, that have a role in supporting the function of neurons in different ways. And one of the glia cells found in our body is called an oligodendrocyte, and this has the function of producing this myelin sheath. So the oligodendrocyte has these kind of membrane extensions that come out and which wrap around the axon of nearby neurons and produces this insulation of the axon. One feature of the myelin sheath that you might have noticed is that it doesn't run along the whole length of the axon. Instead, there are these intermittent gaps in the myelin sheath that have a particular name. They're called nodes of Ranvier. And what happens is that the action potential is occurring specifically at these nodes of Ranvier, these gaps in the myelin sheath, where the axon is exposed to the extracellular fluid. And there's a sense in which the action potential is kind of jumping from gap to gap as it works its way down the axon. And the effect of that is that it moves much more quickly down the axon when the myelin is present compared to when the myelin is not present. So to explain what's happening here, in this diagram we're looking at the axon in yellow with the myelin in purple. We're starting at a part of the axon nearer to the cell body, and the action potential is occurring here to begin with, where we have sodium ion channels in that segment of the axon that is exposed to the extracellular fluid that are allowing sodium to enter the cell during the action potential. The influx of those positively charged sodium ions allows those sodium ions to diffuse within the axon and causes this local current flow, which refers to a flow of positive charge within the axon. And that's enabled by the myelin insulation that by insulating the axon, it allows this local current flow to occur to a greater extent. But that local current flow, that flow of positive charge within the axon, is not strong enough to travel the whole length of the axon. And so instead what needs to happen is that the action potential needs to be regenerated again. And that happens at the next node of Ranvier. So that local current flow causes a depolarization of the axon at the next node of Ranvier, which triggers the opening of the voltage-gated sodium ion channels and allows sodium ions to rush into the cell again. So this is another action potential occurring. And then that triggers local current flow to occur through the myelin insulated area of the axon to the next node of Ranvier. Okay? And so the idea here, the full picture, is that this action potential is happening at one node of Ranvier, causing this local current flow, which then triggers an action potential to happen at the next node of Ranvier, and so on until it gets the signal, works its way down to the synapse. And the effect of this is that action potentials can travel very quickly along myelinated axons, so they can travel at something like up to 100 to 150 meters per second. You may be familiar with multiple sclerosis or MS, which is this neurological condition or autoimmune condition where the symptoms can include difficulty with coordinated movement, problems with balance, sometimes vision problems, sometimes cognitive problems. And this occurs when a person's immune system is attacking the myelin sheath. It's causing damage to this myelin that is surrounding the axon of neurons. And that has the effect of slowing or disrupting the electrical signals that are passing down the axon of the neurons. And you can imagine if the electrical signals that the brain is trying to send to your muscles to coordinate your movement are being slowed and disrupted, that would lead to these symptoms of having difficulty in controlling and coordinating movement. Okay, so the key point here being that myelin insulates axons to increase the speed at which action potentials conduct down the length of the axon. Okay, so are there any questions at this point? Okay. All right, so the first question is to imagine that you have a neuron in a Petri dish in the lab, and you're stimulating the cell body of this neuron with a brief pulse of electrical current. So you're using an electrode here to apply electricity to the neuron. The first time you apply this pulse of electricity, it causes an action potential to be generated in this neuron and to travel down the length of the axon of the neuron. And then you quickly apply a second pulse, but you find that when you apply that second pulse, no further action potential occurs. So which of the following events is likely to have contributed to this? Do you think it's the inactivation of voltage-gated potassium ion channels in the neuron? The potassium ions leaving the neuron during that first action potential that you triggered? Or do you think that it might be because the voltage threshold at which sodium ion channels will open has changed? Okay, so this is a bit more of a tricky question, I guess. So the class is more split across the three options here than compared to the earlier questions. And so my answer here is B. So we talked about the refractory period of the neuron, that there's an absolute and relative refractory period. The absolute refractory period is caused by the inactivation of the sodium ion channels, so sodium ion channels that need to open to trigger another action potential. And so A is maybe a bit of a trick question here. So the reason I'll say that that's wrong is because it's referring to the potassium channels rather than the sodium ion channels. The relative refractory period relates to the hyperpolarization of the neuron that occurs during the action potential, which makes it more difficult for a stimulus to produce another action potential. And that hyperpolarization is due to B, that the potassium ions during the first action potential are moving out of the neuron, which is what causes it to become hyperpolarized inside, to become more negatively charged inside. So that's why I will put B. So C is not quite correct, because while it's true that what matters here for the relative refractory period is the voltage threshold of the sodium ion channels, and how the potential of the cell has moved further away from that threshold when it's in its hyperpolarized state. The actual threshold itself of these channels hasn't changed. So if the threshold at which the sodium ion channel will open is negative 55 millivolts, for example, that threshold won't change regardless of whether the cell is hyperpolarized or not. So it's just that the voltage of the cell has moved further away from that threshold rather than the threshold itself having changed. Okay, so one more question before we go. So here I'm asking you to imagine that you have two neurons in your leg that each have axons that extend from your leg into your spinal cord. If both of these neurons are stimulated at the same time, which signal will reach your spinal cord first? Which of those neurons? Will the signal from both neurons reach the spinal cord at the same time because nerve signals travel at a fixed speed? Does it depend on the length of the axons of those two neurons? Whether that differs between the neurons? Does it depend on whether the axons are myelinated or not for these two neurons? Or is it both B and C? Does it depend on both the length of the axon of each neuron and whether it's myelinated or not? Okay, great. So most people have put D, which is the correct answer. So The idea here is that these electrical signals, it's a physical event. Relating to this opening of ion channels that occurs in a chain reaction down the length of the neuron at these different gaps in the myelin sheath. And it takes time to travel. It doesn't happen instantly. It has to travel from one length of the axon to the other. So if the length of an axon is longer, it will take more time for the signal to travel down that axon. In other words, if your brain is trying to send a signal to your foot, that will take longer compared to if it's trying to send a signal to your shoulder, for instance. So B is correct. C is also correct, as we've just seen, that myelin has this kind of key role in allowing neurons to send electrical signals faster down the axon. And some neurons in our brain and our body are myelinated and others are not myelinated. And that actually means that the latter category are sending signals at a slower speed. So both B and C are correct here. Okay. So so today's content was quite technical and detailed. If you feel like you could use some more kind of revision to sort of consolidate the ideas, we'll be having a kind of hands-on activity in the next tutorial, in the first tutorial, that will hopefully help with that. There's also a video that I've linked here which has some nice animations that show the kind of movement of ions and opening of channels and so on, which I think is a good way of visualizing what I'm talking about, so you might find that useful as well. And next week we'll be building on today's content by talking about the synapse and how this electrical signal that we've been talking about today triggers the release of neurotransmitters in neurons that allows them to talk to each other. Okay, so thank you. I'll see you next week.